Well, thank you very much, Dr. Hermann. Thank you very much to the organizers for having me for this 3R symposium. This is a great symposium that you're organizing here, and in particular, now I think we've all come to realize during the COVID pandemic that there are actually options to kind of broaden the attention to seminars like this by going online. And even though it's not the same as sitting together and having the opportunity to chat after a meeting, it's often a good choice when you want to teach material to a broader audience. So I'm very happy to be part of this, and I'm happy, of course, to share some of my research or some of the things that I've been involved in and have been working on over the last years. I think I try to share my slides and hope that this works well. So you should now see the full presentation of my PowerPoint. Okay. Right. So I've been asked to talk about how to coordinate the 3Rs and the 3Vs in animal research. And now, given the name of this symposium, I assume that you're all familiar with the 3Rs, while many of you may not have heard about the 3Vs or may not know what the 3Vs are. And so I'm going to talk about the 3Vs, which I have coined a couple of years ago as an additional instrument for evaluating for the ethical evaluation of animal research protocols. So I'm going to talk about the role of both the 3Rs and the 3Vs in the ethical evaluation of animal research, but I'm going to focus mostly on the 3Vs as the 3Rs are familiar to most of you. But I will also address potential inconsistencies within and between these two concepts or frameworks, and illustrate how they may be used in ethical evaluation of animal research. So, as I said, I assume that the 3Rs principle are familiar to you. They have been coined almost or more than 60 years ago by now by Russell and Birch, and somewhat unfortunately, Russell and Birch, who wrote this book about the principles of humane experimental technique, they have been more or less reduced to the 3Rs principle over the last decades, and more Russell and Birch is equal to 3Rs and vice versa. Although, of course, their book about the principles of humane experimental technique goes way beyond the 3Rs principles. And a lot of what I'm going to talk about today about the 3Vs, you would also find in their books or in their book and in other researchers' books about the design of experiments and in particular the design of animal experiments. Nevertheless, let's just look at what the 3Rs principle is. It's basically the three questions that every researcher before engaging in an animal research project should ask themselves. And that is, first, is it really necessary to use animals for the aim that I'm trying to achieve with my research project? Or are there alternative methods to using animals, or at least sentient animals? That's the first R, replace. And then if no alternatives are available, the next question would be, is it really necessary to use that many animals, or can I achieve the same objectives by using fewer animals? And finally, refinement. Are there any refinements that would render the procedures that I've planned less harmful in terms of housing conditions, handling, habituating animals to procedures, and if it's about invasive procedures, any form of anesthesia, analgesia, and so forth. So these are the 3Rs, and they have become an internationally accepted approach for researchers to apply whenever deciding to use animals in research and in designing humane animal research studies. and they are used by authorities authorizing animal research, for example, in the EU or in Switzerland, or by institutional animal care and use committees in the US for assessing whether animal research projects are appropriate or whether there is scope for protecting animals or for safeguarding the welfare of the research animals. Now, meanwhile, we've seen huge innovation and development in almost all areas of the 3 Rs, and even though there is still a great need of improving 3 Rs methods, we see increasingly adoption of 3R methods, for example, in regulatory testing of animals, where, for example, in drug development, more and more in silico methods, computer simulations are replacing screening in vivo studies, and increasingly also replacement methods, alternative methods such as organs on a chip or organoid culture models are being used either as a replacement or to complement in vivo studies. And also on the other side, in terms of refinement, which of course is the first choice for all those who do basic research, fundamental research on animals in order to understand animals, that animals are housed under better conditions, that handling methods have been made less aversive, and so forth. All of this is part of a 3 Rs movement across the world, and especially over the last 5 to 10 years, we've seen 3R centers being established almost around the globe, promoting these methods, disseminating them among the researchers, and communicating them to the public. Now, while the 3 Rs are certainly necessary to guarantee humane animal research, they are by no means sufficient to guarantee humane animal research. And that is not least because the core aspect of ethical evaluation is the so-called harm-benefit analysis. Now, ethical evaluation of research projects that involve the use of animals are mandatory both in the United States and in Europe. And this ALAS and FELASA working group on the harm-benefit analysis of animal studies found that both the Guide for the Care and Use of Lab Animals as well as the European Directive require that objectives and benefits of studies using animals need to be weighed against the potential harm inflicted to these animals. And so the harm-benefit analysis is the cornerstone of ethical evaluation, as it will determine if the use of animals can be justified. Now, in fact, a harm-benefit analysis, on the other hand, is just the final stage of the legal principle of proportionality, or a test of the legal principle of proportionality. So constitutional rights law defines 4 stages to a proportionality test, and that can be applied to the assessment of an animal research project. Proportionality tests are conducted in law whenever colliding interests that are regulated at the same level of regulation conflict. So, for example, in animal research, of course, we have on the one hand the freedom of research, on the other hand, the intention to protect the welfare of animals. And so we somehow need an instrument to balance these different interests. And ultimately, a harm-benefit analysis is an instrument to weigh these instruments. But in order to do this properly, we have to proceed along the 4 stages that I've illustrated here. So first of all, of course, any measure needs to have a legitimate aim. And for animal research, these are normally defined by law. So, for example, in the EU directive, all the different purposes for which animal research is a legitimate aim are defined in Article 5 of the directive. where it states that, for example, fundamental research is a legitimate aim. Then, of course, research in view of protecting and safeguarding the health and well-being of animals and humans, but also nature conservation can be a legitimate aim for animal research. Once that's been sorted out, on the next step, it needs to be assessed whether a specific measure, or in this case, a study protocol, is suitable for achieving the intended aim. So suitability in terms of animal research, of course, refers to the scientific validity and reproducibility of a study, the screening of a study protocol for the methods being used and whether they are sufficiently scientifically valid and reproducible in order to achieve the intended aims. And only then, and that is important, as you will see later on, the 3 R's will come into play. The 3 R's, they address the third stage of this proportionality test, where it is assessed whether a particular measure is necessary. So even if there is a legitimate aim and a study protocol may be suitable for achieving a particular aim, it may not be necessary because there may be alternative methods. And so the 3 R's address whether or not it is necessary to use animals or to use procedures that impose harm on animals in the course of a study. And only once these 3 stages are settled and scientific validity and reproducibility have been assessed, or suitability has been assessed on the basis of scientific validity and reproducibility, and necessity has been assessed on the basis of the 3 R's, does it make sense to actually conduct a harm-benefit analysis to weigh the interests between the harms imposed on the animals and the expected benefits of a research project. At this final stage, it is assessed whether a particular study is reasonable and can be ethically justified based on the proportionate measures taken along the study. So ethical evaluation is clearly more than assessing whether the 3 R's have been implemented. And that can easily be seen on this slide, where you see a balance, a harm-benefit evaluation, where on the one hand, we have the harms imposed on the animals. On the other side, we have the benefit of research. And of course, this is not what we want to see. What we want to see is that the benefit of the research outweighs the harms imposed on the animals. Now, in a situation like this, it may happen that even though the 3 R's have been implemented to the full scope, so it has been assessed whether alternatives are available, sample size has been reduced to the absolute minimum, and all refinements possible have been implemented, and yet it still seems that the harms to the animals outweigh the benefits of the research. So the benefits seem too trivial for the weight of these harms to give this study protocol a thumbs up. This makes it evident that the 3 R's alone are not sufficient for ethical evaluation, and more is needed than the 3 R's. Moreover, the 3 R's are just a precondition to harm-benefit analysis, but even in a situation like this, it may happen that a study may not be ethically justifiable. So even though, or even if as few animals are used as possible and the least possible harm is imposed on these animals, unless the results that come out of a study are scientifically valid and reproducible, these animals may have been wasted for inconclusive research. And this is why I put the focus more and more on scientific validity and reproducibility, because besides implementing the 3 R's principle, this is something that has been a bit out of focus by those assessing study protocols for animal research, and something that only in the course of the so-called reproducibility crisis has come more and more in the focus, not only of the scientists, but also of the regulators of animal research. So I assume that you have all heard about the so-called reproducibility crisis, and whether or not you like to call it a crisis, it is certainly clear that there are issues with the scientific validity and reproducibility of science, not just in animal research, but also in animal research. And therefore, I'm going to address some of the issues there are with reproducibility and scientific validity in order to figure out what the causes are for poor reproducibility and what measures can be taken against it. So certainly when working with animals, one obvious cause or potential cause of poor reproducibility is poor validity of the animal model. So if the animal's model being used isn't a valid model of what is being studied, then we should not expect the results to be scientifically valid and reproducible. Now I'm just going to illustrate this with one example that has got some media attention in recent years, and that is the so-called forced swim test. And that is perhaps a particularly good example because it's one where the animal rights movement has basically used the arguments of scientists in order to fight animal research. And the background of this is that this forced swim test has used for a long time in studies on depression and has been used on the one hand as a readout in order to test drugs against depression in animal models, in mice and rats. But it has also been used to induce states of depression in these animals. However, although researchers who have studied the biological construct underlying this test have long warned against using this procedure because they argued that it lacks construct validity, so that it's not a valid biological construct for depressive states in animals, researchers have continued using it as such, and of course by this have become an easy target to animal rights groups who could basically refer to other scientists arguing that this test should not be used in the way it is being used. However, of course, construct validity or the validity of animal models is something that has to be assessed on a case-by-case basis, and there are no common rules by which the validity of animal models can be assessed. But in any case, whenever animal models are being used, then careful validation of these models is a first step before using them on a broader scale. On the other hand, we have problems that are they cover almost the entire field of animal research, and one of those is the use of small sample sizes. Now, of course, sitting in a 3Rs symposium, of course, you may think, well, small sample size is a good thing. It's one of the principles of the 3Rs, reducing sample sizes as much as possible. However, the problem is that if sample sizes are reduced to sizes that are too small, the study may lose its statistical power and may not be suitable anymore to test the hypothesis that is being tested. Now, there have been a number of systematic reviews recently, and these are data from one of them, where Bonapersona and colleagues studied sample sizes and statistical power across the neurosciences and metabolism research. And as you can see here on the left side in this left panel, there is the number of experiments drawn against the statistical power achieved in these studies. And as you can see, the median statistical power achieved across all studies listed here is roughly 18%. Normally, we're aiming for a statistical power of 80%, meaning that if there is an effect to be detected 4 out of 5 studies would actually detect this, while of course with a median statistical power of 18%, 4 out of 5 cases, an effect would not be detected if it were present. And if an effect is detected, then it is likely to be highly inflated, because you need to have a very large effect size if your study is underpowered in order to detect a significant effect. The reason for this is that most researchers don't seem to perform a power analysis or a proper sample size calculation before designing a study, and this can be seen here on the right side of this panel, in the panel D, where you can see the distribution of the number of animals used per study. And as you can see, the highest bar here and the median across all studies is 10 animals per group. And now, of course, 10 animals per group, that sounds more like we've always used 10 animals, so we're going to use 10 animals in the future rather than a proper sample size calculation. Related to this is the problem that in many cases there is a lot of analytical flexibility that researchers have when they do exploratory studies. So they conduct studies and assess lots of different data, and then they may analyze these data in different ways, compare different subgroups of their study populations in order to find interesting relationships or interesting treatment effects. And these are called p-hacking on the one hand and harking on the other hand. P-hacking meaning that multiple statistical tests are performed on the same data set without correcting for multiple testing. And as you can see here, at this dotted line here in the middle is with an alpha value of 0.05, which is the usual level of statistical significance. With only about 15 statistical tests, you will have a 50% chance to find at least one significant finding by chance. So the proportion of type 1 errors, the proportion of finding significant effects when there aren't actually any real effects, increases exponentially with the number of statistical tests being performed. Another problem is harking, and that is that hypotheses get adjusted to the results after the results have been obtained. So harking stands for hypothesizing after the results are known. And we've recently found an interesting case where that we cannot explain other than by harking. So what you can see here is results on the effect of diazepam on the locomotion of mice in an open field test. So this is locomotor activity in an open field test from a systematic review. And there are 150 studies, 151 studies involved in this systematic review. And here on the left side, you can see the effect size with a positive effect size, meaning that the diazepam increased locomotor activity compared to the control group, while negative values mean that diazepam decreased locomotion compared to the control group. Now, interestingly, when we analyzed these data, we found that there is a distribution of effects around the zero. So we have positive effect sizes, negative effect sizes, but the largest density of effects is around zero. So there is in general no overall effect of diazepam on locomotion in the open field test, but a rather widespread to both sides of the null. Interestingly, when we then analyzed the papers for what they had predicted to find in the introduction and method section, we found that those who discovered positive effect sizes, most of them had also predicted to find a positive or so-called anxiolytic effect by diazepam, while those who detected a negative effect size, most of them had predicted to find a sedative or depressive effect of diazepam in the introduction or method section. And then a large proportion of those who did not predict a particular effect, but primarily used locomotion in the open field test as a control measure in their studies, they did not find a significant effect, but found on average zero effect and the distribution around that zero. And we don't see any other explanation of this than harking that the authors just adjusted their hypothesis based on what they found after they had collected their data. And of course, when results are reported like this in the literature, this is going to distort the evidence landscape in the literature, and any systematic review based on these data will then be biased by this biased interpretation of results. And then there are many other risks of bias: randomization, no randomization, no blinding, not defining inclusion and exclusion criteria a priori, and so forth. These are all problems that can distort the effects that are reported in the literature and lead to biased evidence synthesis based on systematic reviews. And finally, rigorous standardization, and that is something that I have more or less added to the list of potential causes of poor reproducibility. And the special thing about standardization is that while all those other causes of poor reproducibility are acknowledged by scientists of bad things that should not be done, rigorous standardization is actually something that is recommended by laboratory animal science textbooks even today. So I need to explain why I consider them as a potential cause of poor reproducibility. And this has to do with the fact that standardization reduces or limits the generalizability of results from animal research. And in order to understand this, we have to go back in history a little bit to the time when reproducibility was introduced as a key principle to establish matters of fact in science. And that was back in the 17th century when the Royal Society of England introduced reproducibility. And from then on, every observation or every fact reported by a scientist had to be replicated independently by an independent laboratory in order to be accepted as scientific evidence. Now, of course, that is no longer a requirement these days. And nowadays, we have more or less replaced independent replicate experiments by within study replicates. So instead of observing just one mouse or one cage of mice, I usually have a whole sample, of animals in a treatment group and a sample of animals in a control group. And the different animals or the different cages of animals within my study design, they are considered the independent biological and or statistical replicates. Now, of course, it's easy to see that the animals within a study, as depicted in this picture, are a lot less independent of each other than the animals would be in independent studies conducted by independent laboratories, by independent scientists. Because all of those animals within a study, they usually share their genotype, they are usually of the same age, they have been housed under the same housing conditions all their life, eating the same food, being cared for by the same care staff, and being tested by the same experimenters. So there's a huge amount of shared genotype and shared environment that, of course, renders study populations highly homogeneous compared to different study populations between independent laboratories. And this is why I consider standardization as a problem for reproducibility, because of course, with this approach, we will obtain highly precise results within studies, but it is highly likely that results of independent studies in different laboratories will be different, because anything in the genotype or the environment of the animals that will be different between different studies may affect the results. And unless we test for the effect of such heterogeneity, we will not know whether our results are specific to our study conditions or whether they are generalizable across different conditions. So I've talked you through what I consider the major causes of poor reproducibility, standing short for poor scientific validity and poor reproducibility. And as you can see, these different causes of poor reproducibility can be mapped onto three aspects of scientific validity, namely construct validity, internal validity, and external validity. Now, construct validity refers to the extent to which a test assesses what it is supposed to assess. And this is what we expect, of course, from an animal model. An animal model should be valid in the sense that it models what we are aiming to model, and this is referred to as construct validity. Internal validity is concerned with the cause-effect relationship, or the trust that we can have into the cause and effect relationship, given the potential of biases. So it is about the validity of a cause-effect relation between treatment and outcome, whether the effects that we measure between a treatment and control group are really due to treatment or might actually be caused or at least biased by some of these problems that I've listed before. And finally, external validity. External validity refers to the generalizability of the results beyond the specific conditions of a study. Generalizability of results to other populations of animals and other conditions of a study. And of course, because independent replication of a study in an independent laboratory depends on external validity, because conditions will never be the same between independent studies, reproducibility is linked to external validity. So based on this, I have proposed the so-called 3Vs principle, because these three aspects of scientific validity are really the core aspects of validity that are relevant when we're talking about scientific validity and reproducibility of animal research. And because there was no similarly, catchy principle as the 3Rs principle, and because the three aspects of validity lend themselves to it, I coined the term 3Vs in analogy to the 3Rs principle. And whereas the 3Rs principle is used to assess the necessity of the use of animals and harm to animals, the 3Vs can be used to assess the suitability of a study protocol in terms of its scientific validity and expected reproducibility of its results before the final ethical evaluation of a study protocol is conducted, the harm-benefit analysis. So I believe that the 3Vs and 3Rs principles, together with the harm-benefit analysis, provide a comprehensive framework for protocol review in animal research. And together with a colleague, Matthias Eckel, we have recently published a paper where we have assessed the internal consistency and compatibility of the 3Rs and 3Vs principle for project evaluation in animal research. And I want to briefly talk you through this paper for the rest of my presentation. So are there any conflicts or are there conflicts between any of the 3Vs? Are there conflicts between construct and internal validity? We came to the conclusion no, because construct validity is about what is being measured, while internal validity is about how it is being measured. So no matter how an animal model is developed, it is important that it is a valid construct. How that construct is being used and how it is being measured that is then a matter of internal validity, and we don't see a reason why these two aspects of scientific validity should get in conflict with each other. Is there a conflict between construct validity and external validity? Here we came to the conclusion, yes, but, and that is because the best animal model, for example, it could be possible that a model is only available in a very specific strain of mouse because it has been developed on a particular genotype. And then, of course, it will not be possible to use other strains of mice in order to assess whether it is generalizable to mice in general or specific to that particular strain of mouse. So external validity may be compromised by choosing the best construct of an animal model. But in any case, what can be done then in order to assess external validity is to complement that specific animal model with the second best model, or to vary other factors that may be varied, for example, the sex of the animals, the age of the animals, the feed or the housing conditions of the animals, in order to assess how externally valid the findings are. And finally, are there conflicts between internal and external validity? There are no direct conflicts, but there may be trade-offs, and these need to be settled by study design. So internal validity often means making results more precise, while external validity means making results more generalizable. And there may be a trade-off in the methods to increase internal validity versus external validity, and finding the right balance between complexity and practicability is actually the art of experimental design. Now, block designs, factorial designs, randomized block designs, they allow standardization within subgroups of animals and heterogenization between groups of animals. And so both aspects, internal and external validity, can be addressed within one study design. And there are good examples for this. And we have recently published a perspectives paper where we explained how we can deal with biological variation in study design of animal research, and specifically recommend using randomized block designs where we can apply control and treatment, or treatment and control conditions in blocks of animals. And these blocks of animals, they can be highly standardized, but different blocks of animals, for example, different strains or animals from different housing conditions, can be included without losing statistical power and actually gaining a lot of external validity and reproducibility by this. Are there any conflicts between any of the three R's? Is there a conflict between replacement and reduction methods? We came to the conclusion no, because replacement is about whether animals are needed in the first place, while reduction is about how many animals are needed if replacement is not possible. Are there conflicts between replacement and refinement? Again, we came to the conclusion no, because if replacement, there has been an argument about methods where replacement is only partial, as for example in the use of fetal bovine serum for as a culture medium for cell culture. So for this serum, calves are killed, and of course this is a welfare issue. But of course, there would only be a conflict if the replaced method, actually the animal method that is going to be replaced by the in vitro method, was less harmful than the replacement method. And this is unlikely to be the case here. And more alternatives exist to the use of fetal bovine serum, so animal-free culture media, and so this is no longer going to be a problem. Finally, is there conflict between reduction and refinement? Yes, there is, and this is actually something that needs to be settled by harm-benefit analysis, because of course the conflict between reduction and refinement is in many ways a question about what do we value more, the life of animals or the number of animals, or the severity of procedures imposed on animals. So the decision between a longitudinal study where repeated measurements are taken from the same animals versus cross-sectional studies where a larger cohort of animals is studied and each subgroup of animals is only measured once. Then whether or not to reuse animals for multiple experiments, that's a similar case. Should always new animals be used for each experiment? Or should we save animal numbers and reuse animals for multiple experiments, leading to a higher burden for those animals? And the similar problem is within subjects versus between subjects designs. For example, if surgical techniques are studied, for example, we could have an experimental treatment on one leg, surgery on one leg with an experimental treatment, and the control treatment with surgery on the other leg. So that would be bilateral surgery. Or alternatively, we could distribute control and treatment surgery to two different animals. So either we have two surgeries on half as many animals, or we have one surgery on each of twice as many animals. And these are trade-offs that for which there is no simple solution and probably have to be assessed on a case-by-case basis in the course of a harm-benefit analysis. And finally, are there any fundamental conflicts between the 3 V's and the 3 R's? Well, frankly spoken, yes, there are, of course. Available replacement methods may not always have the best construct validity. The most refined procedures may not generate the highest construct validity. Reduction of sample size, I've shown you this, may compromise the internal validity, the statistical power of a study. Available replacement methods may produce less externally valid results, and, for example, reduction of animal numbers through standardization may compromise the external validity. So there are many potential conflicts between the 3 V's and the 3 R's. However, that isn't really a problem, because in that proportionality test, if you remember, the 3 R's, they come secondary to the 3 V's. And of course, the 3 R's principle isn't an end in itself, but it is something that is applied to a particular study protocol. And of course, we have to make sure that the study protocol is suitable, that is, it is scientifically valid and produces results that are likely to be reproducible. And only then does it make sense to assess how the 3 R's have been implemented. So, of course, we will always first have to define the study protocol, make sure it is scientifically valid, and then apply the 3 R's. And if there are still conflicts that remain, or if alternative scenarios are there that should be evaluated, they need to be evaluated in terms of harms and benefits at the level of the harm-benefit analysis. Now, with this, I'm going to skip the last 2 slides, but would like to thank you for your attention and refer to some of the members of my lab who contributed in one way or the other to some of the research that I've shown you today. In particular, Bernhard Völkel, who has been extremely important in developing my ideas about the relationship between standardization, external validity, and reproducibility further, as well as some of my long-standing collaborators, especially Joseph Garner from Stanford, Malcolm MacLeod from Edinburgh, and Yoav Benjamini from Tel Aviv. And of course, without generous support from all of these different funders, none of this would ever have been possible. So thank you very much for your attention, and I'm happy to take any questions. I didn't actually have a watch to check my time, so I have no idea how much time, or if we do have any time left for discussion.